My name is Ali Tehrani. I'm the President and CEO of Zymeworks. Zymeworks is a biotechnology company based in Vancouver, British Columbia, Canada. We are in the business of developing best-in-class antibody therapeutics in the areas of cancer, autoimmunity, and inflammatory diseases. And we are delighted to be here at the Bispecific Summit. It's been a great conference so far. Certainly learned a lot, and it's very humbling to see what other people are doing.
Should you talk more about your pipeline? Could you
Sure.
talk me through it?
So one of the things that came up at this conference was that there are about 35 different bispecific formats. And everybody wants to know what everybody else's format is and how does it differ, what kind of advantages it offers. Our bispecific format is a IgG1 format with two different heavy chains. So it's a heterodimeric IgG1 with specifically having protein engineering done in the CH3 domain. We do not use any linkers or any modifications or, any attachment of other domains. It's pure protein engineering of the CH3 domain, and we're currently working on heterodimerizing the light chain as well. Most people resort to a common light chain approach. We are working on heterodimerizing the light chain as well. So ultimately, what you're left with is a full-length antibody that does not involve, as I said, any linkers or any addition of any other domains. And an additional advantage is the fact that our antibody comes with tailored effector function. What that means is that we've got complete control over the CH2 domain such that we can modulate binding to the Fc gamma R receptor, whether it's upregulating it or downregulating it, which becomes very important in inflammatory and autoimmune diseases. Moreover, the last feature is that our format comes with optimized serum half-life, which what we're really talking about here is binding to the FcRn receptor. So we've kept that intact, and in some respects we've optimized it. Besides this bispecific format, we're also putting the finishing touches on a antibody alternative protein scaffold, which is multivalent. So here, most people talk about bispecifics, which means binding to two epitopes or two targets. Our antibody alternative is capable of binding to more than two targets and two epitopes. I'm not at liberty to say how many, but we can leave it as 2+. It's smaller than an antibody, and it has IgG1-like binding properties, similar binding to FcRn as IgG1 does. So its serum half-life is exceptional. What this does is enables us to have a couple of solutions for different biologies, which is another theme that came out of this conference, is that you have all these formats, and you perhaps need all these formats for the different biologies that are required to build the different drugs. So we have an antibody solution, and we have an antibody alternative solution. As far as the drug development pipeline at Zymeworks using these formats, we're currently working on our own preclinical programs, which we hope to announce and let everybody know about what we're working on in the next little bit. In this line of work, you never want to jump the gun, come out too early and hand wave, because the bar is very high, and the expectations are that you got to really have enough meat on the bone, or have a good solid story. You just can't come and say, well, I'm intending on doing this, or I've got these dreams, because these are a tough crowd.
Yeah, they're a tough crowd. And so, can you give any idea of the timeline of when you'll be entering phase 1 with any of your candidates?
Well, part of our strategy is that we're looking for strategic partners, what we call collaborative drug development. I don't think at this moment we're making getting into the clinic a priority of ours. What we want to do is work with partners such that they have ideas of targets or biologies and they want to use our formats, and together we combine to have a product that goes into the clinic. And this is really the story behind our recently announced deal with Merck. Really what happened there is that Merck came with certain targets. We came with a proprietary bispecific format or a scaffold. You know, we're working together to generate next generation of best-in-class therapeutics that ultimately Merck will take into the clinic. We're not drug developers, we're protein engineers. Companies like Merck are drug developers. They're excellent drug developers. So we want to stay as close as possible to our core competency and let others who are really good at what they do apply their expertise and mitigate the risk. That is not to say that in the future we won't pursue our own programs for going into the clinic, but at this moment, probably for the next couple of years, we're more focused on strategic partnerships where our technology is utilized for developing what we go into the clinic.
Could we talk a little bit more about the Merck partnership and actually get kind of an idea of what it looks like? Are there milestones associated to it? And
Absolutely, absolutely. So it's your typical biotech deal. What we've done is we have certain preclinical milestones which really validate the biology of the product. And part of that is validating the format, the scaffold as well, relative to the biologies. And then after that, they take it into the clinic and there's certain milestones associated with that, and at the very end, you have commercialization milestones. So it's your very typical deal. But I think the exciting thing here is the Merck team is not relying on us to do all the work. Some of your biotech deals basically is a small company has an idea of a molecule with a target and a scaffold, and someone else pays for it till it gets to commercialization, and they say, thank you very much, I'm going to take it from here.
Yeah.
In this situation, it is really a collaborative drug development. You know, the Merck expertise, the Merck knowledge and their targets, our bispecific format married together, working together to really develop and generate a top-notch product that ultimately has the highest chance of success in the clinic.
Yeah. And so you're looking for other partnerships now?
Yeah, our model has always been, look at strategic partnerships. So we're always on the lookout for good partners. Merck is an excellent, excellent partner, and we might end up doing only deals with Merck, but this is business, and you always have to, make sure that you leave your options open.
Sure. And then we spoke earlier about you just getting into bispecifics relatively recently, 3 years ago. Could you comment a little bit about the field now and why is it exciting and what does the near future hold?
This conference started with an excellent talk by Janice, and what she said is she provided some data that said antibodies are very hot and bispecifics are the hottest areas in the antibody world. And the antibody drug conjugates is the next wave right behind the bispecifics. In your conference, it's very evident that the field is very hot because you see to your left and right all decision makers from every major company. Every major company is here. And the people that are here are the CSOs and the VP R&Ds. Actually, it'd be interesting to see some sort of a graph to see how many C-levels or senior executives attended versus a principal scientist. And I bet you'll see a ratio which is very much skewed towards the senior levels. That shows you that they care and they want to know what the competitors are doing or learn something that they could help their own programs. It is a very hot area, but it's a small field because it's very difficult, it's very complex. It's not something that smaller companies can readily get into. Not too long ago, binding affinity was the issue that everybody was focusing on, but it was a field that everybody could get into. You could be 4 or 5 man operation and get in and try to solve something with binding affinity, but binding affinity is not that difficult anymore. anymore.
Yeah.
Bispecifics are extremely complex, and there are no leaders in the field. So there's an opportunity for someone to get ahead of the race.
Yeah, it's true.
You know, my last point for you is, and for your organization, is really good job. This was a great conference. I can tell you for sure that we came with a sense of skepticism. We didn't know what it's going to be like. Is it going to be a waste of time? Because most of us are traveling from, you know, big distances and far away. But I'm pleasantly surprised. I took a lot of notes and I learned a lot and met a lot of good folks. It was very well organized. I think one of the things that I liked about it is the organizers stayed out of our way. Some other conferences, the organizers are right in your face all the time and wanting to promote something or promote some sponsor. This was as scientific in the industry as one could expect without, you know, just bothering you. Because those are the ones that you don't want to go back to. Like, oh, please don't sell me. I know who you are. Stop selling me. So it was very well done. So kudos to the organizing committee, and I'm excited to go to the conjugate one too by you guys.
Cool.